Welcome to clickmeproject.com. Now we are going to see the paper, a link analysis extension of correspondence analysis for mining relational databases. Traditional statistical, machine learning, pattern recognition, and data mining approaches usually assume a random sample of independent objects from a single relation. Many of these techniques have gone through the extraction of knowledge from data, almost always leading in the end to the classical WNG tabular format containing features for the sample of the population. These features are therefore used in order to learn from the sample, provided that it is representative of the population as a whole. The focus of the techniques have moved over from the analysis of the features describing each instance belonging to the population of interest to the analysis of the links existing between these instances in addition to the features. This paper proposes a link analysis based technique allowing to discover relationships existing between elements of a relational databases or more generally a graph. More specifically, this work is based on a random walk through the database defining a Markov chain having as many states as elements in the database. Suppose, for instance, we are interested in analyzing the relationships between elements containing in two different labels of a relational databases. In this process, a two-step procedure is developed. First, a much smaller reduced Markov chain only containing the elements of interest, typically the elements contained in the two tables, and preserving the main characteristics of the initial chain is extracted by stochastic complementation. An efficient algorithm for extracting the reduced Markov chain from the large sparse Markov chain representing a database is proposed. Then the reduced chain is analyzed by, for instance, projecting the states in the subspace spanned by the right eigenvectors of the transition matrix called the basic diffusion map in this process, or by computing a kernel principal component analysis on a diffusion map kernel computed from the reduced graph and visualizing the results. Indeed, a valid graph kernel based in the diffusion map distance extending the basic diffusion map to directed graphs is introduced. Next we see the flow. First, we get the protein sequences and the interaction datasets. Protein-protein interaction occurs when two or more proteins bind together, often to carry out their biological function. Many of the most important molecular processes in the cell, such as DNA, replication are carried out by large molecular machines that are built from the large number of protein components organized by their protein-protein interaction. Interactions between proteins are important for the majority of biological functions. We collect malfunctioning protein-protein interaction dataset and store it into database. Then we extract the sequence from the database. Then apply the protein malfunction process. Many interactions of proteins are maybe connected improperly or abnormally interacted or repeatedly interacted. In identification of protein malfunction, we identify and destroy these proteins from the original protein interactions, and remaining original interactions are stored into database. Then the subgraph of this process is constructed for these final interacted proteins. Next one is stochastic complementation. The concept of stochastic complementation is applied to the analysis of the graph through the random walk on a graph model. From the initial graph, a reduced graph containing only the nodes of interest and which is much easier to analyze is built. Analyzing the relationship between two sets of nodes, a reduced Markov chain can be computed from the original chain. The set of states is partitioned into two subsets, S1 and S2, where S1 is corresponding to the nodes of interest to be analyzed and S2 is corresponding to the remaining nodes to be hidden. The resulting reduced Markov chain obtained by censoring the states S2 is the stochastic complement of the original chain. Thus, performing a stochastic complementation allows focusing the analysis on the tables and the elements representing the factors of interest. The reduced chain inherits all the characteristics from the original chain. It simply censors the useless states. The next process is diffusion map. A diffusion map is a machine learning algorithm for dealing with dimensionality reduction. In this process, we use KDM-PCA. The proposed two-step procedure can be considered as a generalization of correspondence analysis. The relationships between the attributes of the two categorical variables are usually analyzed through the biplot, a 2D representation of the attributes of both variables. The coordinates of the attributes on the biplot are obtained by computing the eigenvectors of a matrix. Multiple correspondence analysis are the extension of simple correspondence analysis to a large number of categorical variables. The corresponding graph is built naturally by defining one node for each individual and for each attribute, while a link between the individual and an attribute is defined when the individual possesses this attribute. This configuration is known as the star schema in the data warehouse or relational database fields. Finally, we display the reduced Markov subgraph and the reduced interactions after applying the KDM-PCA process. Next, we see the running procedure. First, open the NetBeans. Then open our project into NetBeans. Then add all jar files into libraries. Then just right click load.java and click run file. First select the interaction dataset. Click open. All dataset informations are stored into database. Then select the sequence dataset. Click open. Okay, the sequence informations are inserted into database. These are the protein interaction and the sequence informations. Then click identification. First, we eliminate the missing interactions. Click eliminate missing. Totally, we have 19 interactions and missing interactions are 3. We remove the 3 interactions. So finally, we have 16 interactions. Then click malfunction. First, display the sequence and identify malfunction from the sequence. These 3 are the malfunction in sequence. So we remove these 3 from the dataset sequence. So finally we have totally these sequences. Okay. Then click graph button. Click draw. These are the interactions from the proteins. The sequence interactions. Then click stochastic complementation. First we calculate the adjacency matrix. Then click the transition matrix for each sequences. Then click diagonal matrix. Okay. Then the Laplacian matrix. Okay. Next one is matrix partition. Using these values, we partition the matrix. Okay. We split the matrix into 2, that is state 1 and state 2. The next one is matrix representation for these 2. Then next one is reduced states. Okay. So we display the reduced chain. These are the reduced chain. The next process is PCA. In this PCA, we have calculated eigenvectors, correlation matrix, and finally we display the final states. That is the reduced states using PCA. Then click reduced one. This is the final states using PCA. Then click the view button. In this view option, we select each protein and click view button. We display all information related to that protein. This is a protein image, the protein structure, protein function, and this is affected by the protein. Thanks for listening.